Good day, everyone. So, my report is about microbial metabolism fueling cell growth. So, a glimpse of history. So, in the 1850s, Louis Pasteur tried to determine how alcohol develops from grape juice. Biologists had already noticed that when grape juice is held in large vats, alcohol and carbon dioxide are produced and the number of yeast cells increases. They concluded that the multiplying cells were converting sugar in the juice to alcohol and carbon dioxide. Pasteur agreed, but could not convince two very powerful and influential German scientists named Justus von Liebig and Friedrich Euler, who refused to believe that microorganisms caused the breakdown of sugar. Both men mocked the hypothesis, publishing pictures of yeast cells looking like miniature animals taking in grape juice through one end and releasing carbon dioxide and alcohol through the other. Pasteur studied the relationship between yeast and alcohol production using a strategy commonly employed by scientists today. That is simplifying the experimental system so that relationships can be more easily identified. First, he prepared a clear solution of sugar, ammonia, mineral salts, and trace elements. He then added a few yeast cells. As the yeast grew, the sugar level decreased and alcohol level increased, indicating that the sugar was being converted to alcohol as the cells multiplied. This strongly suggested that living cells caused the chemical transformation. Liebig, however, still did not believe that the process was occurring inside microorganisms. To convince him, Pasteur tried to extract something from inside the yeast cells that would convert the sugar. He failed, like many others before him. In 1897, Edward Bonner, a German chemist, showed that crushed yeast cells could convert sugar to ethanol and carbon dioxide. We now know that the cell's enzymes carried out this transformation. For these pioneering studies, Bonner was awarded a Nobel Prize in 1907. He was the first of many investigators who received a Nobel Prize for studies on the processes by which cells degrade sugars. So, microbial metabolism. So, microbial cells, as well as all other cells, need to accomplish two fundamental tasks to grow. They must continually synthesize new parts, such as cell walls, membranes, ribosomes, and nucleic acids, that allow the cell to enlarge and eventually divide. In addition, cells need to harvest energy and convert it to a form that can power the various energy-consuming reactions, including those used to make new parts. So, the sum of all chemical reactions in cell is called metabolism. So, metabolism is important to humans for a number of reasons. For example, as scientists look for new supplies of energy, some are investigating biofuels. Biofuels means fuels made from renewable biological sources, such as plants and organic waste. Microbes can produce biofuels, breaking down solid materials such as corn stalks, sugarcane, and wood to make ethanol. Microbial metabolism is also important in food and beverage production. So, in the laboratory, products characteristic of specific microbes can be used as identifying markers. In addition, the metabolic pathways of organisms such as E. coli serve as an important model of studying similar pathways in eukaryotic cells, including those of humans. Finally, microbial metabolism is medically relevant because metabolic processes unique to bacteria and potential targets for antimicrobial medications. So metabolism can be separated into two components, catabolism and anabolism. Catabolism is the set of chemical reactions that degrade compounds, releasing their energy. Cells capture that energy and use it to make ATP. So ATP is the energy currency of the cell. And anabolism, on the other hand, or biosynthesis, is the set of chemical reactions that cells use to synthesize and assemble the subunits of macromolecules, using ADP for energy. So as described in chapter 2, the subunits of macromolecules include amino acids and nucleotides, monosaccharides, and fatty acids. Although catabolism and anabolism are often discussed separately, they are intimately linked. As mentioned, ATP made during catabolism is used in anabolism. In addition, some of the compounds produced during catabolism are precursor metabolites, which represent an intersection of catabolic and anabolic processes. So this is the picture of how the process is made. So next is the harvesting energy. So energy is the capacity to do work. It can exist as potential energy or stored energy, and kinetic energy are also known as energy of motion. So potential energy can be stored in various forms, including chemical bonds, a rock on a hill, or water behind a dam. Energy is the universe. Energy in the universe can never be created or destroyed. However, it can be changed from one form to another. In other words, although energy cannot be created, potential energy can be converted to kinetic energy and vice versa. And one form of potential energy can be converted to another. Hydroelectric drums, so this is the example. Hydroelectric dams release the potential energy of water stored behind a dam, creating the kinetic energy of moving water. This can be captured to generate an electrical current, which can be used to charge a battery. So the potential energy here is the water behind that dam. So if the water behind that dam will be released from being stored, so the water will flow, and that is now we call kinetic energy, because the water is now moving and can be captured, so we can generate an electrical current. And that is what it means by potential energy can be converted to kinetic energy. So metabolism involves energy transformation processes. Cells take energy of one form and convert it into another. For example, photosynthetic organisms harvest the energy of sunlight, using it to power the synthesis of organic compounds from carbon dioxide. By doing so, they convert the kinetic energy of photons. So photons, that is the particles that travel at the speed of light. So energy of protons to the potential energy of chemical bonds. And chemoorganotropes obtain energy by degrading organic compounds. They then use some of that energy to make other organic compounds. So in other words, they take the potential energy of certain chemical bonds and use it to create other ones, because chemoorganotropes depend on a constant source of organic compounds. So They generally rely on the metabolic activities of photosynthetic organism. So the example here is this. You can see it in the picture. So the radiant energy is from the sunlight, and the photosynthetic organism here is the rice grain, and it harvests the energy of sunlight and use it to power the synthesis of organic compounds from carbon dioxide. So this converts the radiant energy to chemical energy. So the chemoorganotrophs here is the rat. So chemoorganotrophs degrade organic compounds, harvesting chemical energy. So the rat has now the energy to do work. So the amount of energy released by breaking down a compound can be explained by the concept of free energy. So the energy available to do work from a biological perspective, this is the energy released when a chemical bond is broken. In a chemical reaction, some bonds are broken and others are formed. If the starting compounds have more free energy than the products, energy is released. The energy is exergonic. In contrast, if the products have more free energy than the starting compounds, the reaction requires an input of energy, and that is called the endergonic. So as what you can see in the peak, the ball there is an example. Okay, let's zoom. Okay. So exergonic reaction have more free energy because the energy is released in reaction. Di ba? Gikan mo siya sa taas and it rolled down deliberately. And uh, on the other hand, endergonic reaction there requires input of energy because gapaangat man siya. So, next is the components of metabolic pathways. The series of chemical reactions that converts a starting compound to an end product is called a metabolic pathway. Pathways can be linear, branch, or cyclical. Like the flow of rivers controlled by dams, their activities can be adjusted at certain points. Wait. Okay. So, this allows a cell to regulate certain processes, ensuring that specific molecules are produced in precise quantities when needed. So, if a metabolic step is blocked, products downstream of that blockage will not be made. So, role of enzymes. So, an enzyme is a molecule, usually a protein, that functions as a biological catalyst, speeding up the conversion of one substance. The substrate into another, the product. A specific enzyme facilitates each step of a metabolic pathway. Without enzymes, energy yielding reactions would still occur, but at rates so slow they would be insignificant. So, an enzyme catalyzes a chemical reaction by lowering the activation energy. So, activation energy, this is the energy it takes to start a reaction. Even an exergonic reaction has an activation energy, and by lowering this barrier, an enzyme speeds the reaction. So next, the role of ATP. So adenosine triphosphate, or ATP, is the main energy currency of cells, serving as the ready and immediate donor of free energy. So the molecule is composed of ribose, adenine, and three phosphate groups arranged in a row. Cells produce energy currency by using energy to add an inorganic phosphate group, or PI, to adenosine diphosphate, or ADP, forming ATP. That energy currency can then be spent by removing the phosphate group, releasing energy and converting the molecule back to ADP and phosphate group, or PI. So cells constantly produce ATP during exergonic reactions of catabolism, and then use it to power endergonic reactions of anabolism. So continuing, precursor metabolites. So certain intermediates of catabolic pathways can be used in anabolic pathways. So these intermediates, intermediates or precursor metabolites. These serve as carbon skeletons from which subunits of macromolecules can be made. For example, the precursor metabolite pyruvate can be converted to any one of three amino acids: alanine, leucine, and valine. So it's on the table. So pyruvate. See the proteins of the amino acids alanine, leucine, and valine. So moving forward, enzymes. So recall that enzymes are biological catalysts which increase the rate at which substrates are converted into products. So several environmental factors influence how well enzymes functions. Each enzyme has a narrow range of conditions, including temperature, pH, and salt concentration, within which it functions best. A 10 degrees Celsius rise in temperature approximately doubles the speed of enzymatic reactions until optimal activity is reached. Where am I again? So this explains why bacteria tend to grow more rapidly at higher temperatures. So if the temperature is too high, however, proteins will denature and no longer function. So most enzymes function best at low salt concentration and pH values slightly above 7. Okay, so moving forward, fermentation. So fermentation is used by organisms that cannot respire, either because a suitable or inorganic terminal electron acceptor is not available, or because they lack an electron transport chain. So E. coli is a facultative anaerobe able to use any of three ATP generating options. These are aerobic respiration, and fermentation. In contrast, the only option for Streptococcus pneumoniae is fermentation because it does not have an electron transport chain. So the end products of fermentation are significant for a number of reasons. For one thing, certain end products help identify bacterial isolates because a given organism use a characteristic fermentation pathway. In addition, some end products are commercially valuable. So in the previous chapter, it describes how foods and beverages are produced using fermentations. Important end products of fermentation pathways include lactic acid, ethanol, butyric acid, puponic acid, 2,3-butanoidyl, and mixed acid. So that's the important end products of fermentation. So next is the photosynthesis. So plants, algae, and several groups of bacteria harvest the radiant energy of sunlight and then use it to power the synthesis of organic compounds from carbon dioxide. The capture and conversion of radiant energy into chemical energy is called photosynthesis. So the general reaction with X indicating an element such as oxygen or sulfur is as follows. So light energy, so you can see this. So photosynthetic processes are generally considered in two distinct stages. The light reactions, also called the light-dependent reactions, capture radiant energy and convert it to chemical energy in the form of ATP. An unrelated set of reactions, sometimes called the light-independent reactions of dark reactions. So uses the ATP to synthesize organic compounds. This involves carbon fixation, which will be covered in a separate section. So carbon fixation. So continuing. So chemoautotrophs and photoautotrophs use carbon dioxide to synthesize organic compounds. The process of carbon fixation in photosynthetic organisms, this process is called the light-independent reactions. Carbon fixation consumes a great deal of ATP and reducing power, which should not be surprising considering that the reverse process or oxidizing those same compounds to carbon dioxide liberates a great deal of energy. So the Calvin cycle is the most common pathway used to fix carbon, but some prokaryotes incorporate carbon dioxide using other mechanisms. For example, the green bacteria and some members of the archaea use a pathway that reverses the steps in the TCA cycle. Okay, so the Calvin cycle or Calvin-Vinson cycle, named in honor of the scientist who described much of it. So it's a complex cycle. So the easiest way to understand the outcome is to consider 6 turns of the cycle. Together, these turns incorporate 6 molecules of carbon dioxide and produce the equivalent of 1 molecule of fructose 6-phosphate and an intermediate of glycolysis. So the Calvin cycle has 3 essential stages. So in stage 1, carbon dioxide is added in ribulose 1,5-biphosphate to start a new bond of the cycle. So this is the incorporation of carbon dioxide into an organic compound. And stage 2, ATP and ADPH are used to reduce the product of stage 1, reducing glyceraldehyde 3-phosphate, which can be used in biosynthesis. So in stage 2, this is the reduction of resulting molecule. And the stage 3 is the ribulose 1,5-biphosphate is regenerated so that the cycle can continue. So in stage 3, regeneration of the starting compound. So next, anabolic pathways synthesizing subunits from precursor molecules. So lipid synthesis of most lipids requires fatty acids and glycerol. To produce fatty acids, the acetyl group of acetyl-CoA or the precursor metabolite produced in the transition step is transferred to a carrier protein. This carrier holds the developing fatty acid chain as two carbon units are added. So when the fatty acid reaches its required length, usually 14, 16, or 18 carbon atoms long, so it is released. The glycerol component of the fat is synthesized from the precursor metabolite, the hydro, say acetone phosphate, which is generated in glycolysis. And amino acid synthesis, so proteins are composed of various combination of usually 20 different amino acids. So these amino acids can be grouped into structurally related families that share common pathways of biosynthesis. And glutamate so this is the one of the amino acids, the glutamate. So amino acids are necessary for protein synthesis, but glutamate is especially important because it provides a mechanism for bacteria to incorporate nitrogen into organic material. And aromatic amino acids, so synthesis of aromatic amino acids such as tyrosine, phenylalanine, and tryptophan requires a multi-step branching pathway. So this serves as an excellent illustration of many important features of regulation of amino synthesis. And lastly, nucleotide synthesis. Nucleotide subunits of DNA and RNA are composed of 3 units, 5-carbon sugar and a phosphate group and a nucleobase, either a purine or pyrimidine. So they are synthesized as ribonucleotides, but this can be converted to deoxyribonucleotides by replacing the hydroxyl group on the 2 or 9 carbon of the sugar with a hydrogen atom. So that's all. Thank you.